Good morning, good afternoon for some of you, and for some of you, even good evening. Welcome to our webinar titled Chronic Wasting Disease Spillover Preparedness and Response. Thank you for joining. I'm Mike Osterholm. I direct the Center for Infectious Disease Research and Policy at the University of Minnesota, the organization that helped put this various report together here. Joining me today, though, are 5 speakers who are really among the very top scientists in the world, among the practice aspects of CWD that are so very important. They've all served as co-chairs for the 5 working groups, and I'll get into that in just a minute. We have with us today Dr. Russ Mason, Dr. Glenn Telling, Dr. Sabina Gilch, Dr. Debbie McKenzie, and finally Dr. Brian Appleby. And we will go through the 5 working group findings today to actually share with you what we found and what that means. If I could have the next slide. Thank you. So I'll give a brief introduction overall, but then we'll go into each of the 5 work group areas. Each actually was a chapter in our report. The first one was CWD management and surveillance in wild and captive cervids. Chapter 2 is CWD diagnostic laboratory testing in animals. The third one was spillover to non-cervid production animals. The fourth one is environmental implications of carcass and contaminated item disposal. And the final one was detecting CWD spillover into humans. We will then, after hearing from each of the 5 co-chairs and these various 5 groups, we'll then break into a question and answer session that you'll be able to provide your question by putting it into the chat that'll be monitored. And Dr. Corey Anderson will then work with our 5 panelists to come up with the answers. And then we'll close from there. Next slide, please. Now, in terms of the issue of CWD, we really believe there are multiple factors right now that have contributed to the need for this project. First of all, as you all are so well aware, there's a continued spread of CWD in wild cervids. It's been remarkable. The potentially evolving CWD prion strains is a reality. And so combine that with the continued spread, there's more and more chances for this to happen. With the accumulation of CWD prions in the environment, we also know that that too enhances transmission. Continued exposure to humans who consume or interact with cervid products is a reality. We find in many instances situations where people knowingly are consuming the venison even when it is positive for CWD. And up to this time, there really has been a lack of a multidisciplinary approach. And what this effort was really meant to try to do, and I think we've achieved that, is bring together experts from all the different areas that possibly could be involved or should be involved with this particular challenge. Next slide. Just to give you an idea what the working group process was like, first of all, in the summer of 2023, we identified 5 CWD focus areas that we believed needed to be addressed. In August of 2023, we named 10 co-chairs as well as 58 members to the various working groups, a simply remarkable collection of the best and the brightest in this area. And then from November of 2023 to August of 2024, we held 25 different working group meetings, over 35 hours of discussion. And from that evolved the report, which you'll hear the summary of today. Finally, in January of this year, we actually published Chronic Wasting Disease Spillover Preparedness and Response, Changing at Uncertain Future, Charting an Uncertain Future. And today we'll make sure that you have access to that report if you haven't seen it before, but you're going to also get a summary of what it was all about. Next slide. So here are some of the biggest takeaways that we found. Unclear how CWD spillover into humans or any other non-cervid species would appear. There's no CWD-specific surveillance in non-cervid animals. There's no single test capable of identifying a spillover. No standardized surveillance of CWD strains, and frankly, our lab capacity is limited and difficult to expand. Next slide. I now want to move over to the first working group area. This was co-chaired by Dr. Russ Mason and Dr. Colin Gillen, and it was a remarkable set of discussions about where this is at, where it's going. You can see the other members of the working group here. And with that, next slide. I want to just add that if you look at the CWD management and surveillance in wild and captive cervids, approaches to CWD surveillance and management across North America are very significant. Wildlife agencies are challenged to maintain hunter engagement of CWD surveillance, largely owing to disease fatigue. Currently, there is really no methods are available for this area. And with that, I'm going to turn it over to Russ Mason to continue with the rest of this presentation. Russ?
Thank you, Mike. Before we move to this slide specifically, I just want to mention that we took pains to create a working group that was broadly representative of experts in the field. So 4 state agencies were represented: Michigan, Minnesota, Wisconsin, and Oregon. The Native American Fish and Wildlife Society was present. The Southeastern Cooperative Wildlife Disease Study was represented, as well as the Wildlife Management Institute, the National Deer Association, and Michigan State University. Now, moving to this slide, perhaps the most important part from my perspective is that mostly as a result of no dedicated funding, different states have very different surveillance strategies and intensities. There's an Appendix A to the report where we provide data that we collected through a survey administered through the Association of Fish and Wildlife Agencies. So nationally, if you look across states, the number of animals sampled ranged from 0 to 36,000 roughly, but in CWD positive states ranged from 199 animals to the 36,000. And in CWD negative states, sampling ranged from 0 to 3,250. So again, perhaps as a result, mostly as a result of lack of dedicated funding, states have very different sampling strategies, and there have been no efforts to quantify the relative effectiveness of various strategies. And apart from culling, no attempts to evaluate the utility of management interventions. After decades of experience, most hunters, as Mike mentioned, appear to be ignoring or minimizing CWD messages. And of course, this presents a real challenge for agencies as they try to reach out to the hunting community and get them to cooperate with strategies that they devise. And as Mike again mentioned, in some instances where surveys have been done, hunters that receive a positive test go ahead and eat and share the venison anyway. And it's not just some. In at least one case, it was the majority of hunters that were surveyed. And finally, continentally, CWD appears to be spreading unaffected, more or less, by management intervention. Next slide, please. There's almost no information about the geographic distribution of CWD strains in the wild, and no attempts to evaluate whether different strains are more or less infectious or potentially zoonotic. This is important from an agency point of view. At least when state agencies sample, they're talking about CWD writ large. There's very little information about different strains that may be circulating and the different impacts those strains might have. And again, in Appendix A, there's some information there to suggest that this is the case, as well as the fact that because of lack of funding, states have very limited options when it comes to actually performing tests. As part of the survey, we asked states to tell us whether, where they tested animals and would they be able to accommodate a surge above what they're doing now, say 10,000 animals more. What we found was that only 4 states had existing capacity to collect and evaluate greater than 10,000 samples. Only 10 have the capacity to collect and evaluate 6,000 more samples. And one state indicated that it would refuse to collect additional samples because of biosecurity concerns, specifically staph infected by other deer transmissible diseases. When we asked states, did they have a backup lab, any kind of plan if the lab that they worked with was unable to service their needs, we found that no state had a formal arrangement for testing with more than a single laboratory. And importantly, the majority of states relied on just 4 laboratories for testing. So 4 laboratories are doing the majority of testing in the United States. Next slide, please. All available information indicates that CWD poses an existential threat, or at least to some, or at least the ones that have been studied, to some populations, perhaps all populations in North America. As CWD continues to spread and increase in prevalence, unintentional human and livestock exposures must also be increasing, and this likely increases the probability of disease spillover into perhaps genetically predisposed individuals. Emerging evidence also shows that non-cervid free-ranging species, for example, feral hogs, are competent vectors for CWD. and another potential source of exposures for humans and uncervid livestock as well. Eurasian boars likely represent a competent source of transmission among captive cervid facilities. Yet even opportunistic CWD sampling of non-cervid wildlife is, for any practical purpose, virtually non-existent. And with that, I'll turn it back to Mike.
Thanks, Russ. Outstanding presentation and all the work that you did along with Colin. Now we're going to move to the Prion Biology and Diagnostics Working Group, which was co-chaired by Glenn Telling and Byron Coey. Glenn is going to be presenting today. Glenn is the director of the Prion Research Center at Colorado State University, where he leads research on prion disease mechanisms and the CWD species barrier. Thank you, Glenn. We're glad to have you with us, and the floor is yours.
Okay, Mike, thanks a lot. If I could have the next slide. So our group, so I'm representing our group, which was co-chaired with Byron Coey, and it was a very well-represented international group of folks with interests and expertise, mainly in the basic research of prion biology. I think it's worth pointing out that this is an infectious disease that is part of a unique group of infectious agents, the prion diseases, which include also, in addition to chronic wasting disease, scrapian sheep, bovine spongiform encephalopathy in cattle, which was a major epidemic global epidemic in the 90s and early decades of this century. And human forms, most prominently chronic wasting disease. And so the issue here with the biology of these diseases, which is unique, involves the extremely unusual mechanism of replication and transmission. These are not conventional pathogenic agents like viruses, bacteria, fungi. These are, in fact, proteins. Devoid of informational nucleic acids. Moreover, these are proteins that are encoded by the host species itself. And so the mechanism of infectious transmission involves the conformational conversion of this normal protein, which everybody who's on this webinar expresses, predominantly in the central nervous system. All mammals express this. But it involves the conformational conversion of this normal protein into an infectious form. So this is a revolutionary idea for which there have been several Nobel Prizes already awarded. The challenge is to understand the basic mechanisms of conformational conversion. And I think it's safe to say that the biology of these diseases presents extreme challenges for investigation. The latency period of these diseases is extremely long, both naturally and under experimental conditions. And the unusual features of the infectious agent present major obstacles for rapid discovery. So this is where we are in 2025. I think it's really important to point out that my co-chair, Byron Coey, has really pioneered more recent work on CWD diagnostics, which have involved, because this is an infectious process, we can use the biology of this conversion as a means of amplifying very small amounts of infectivity. It's unfortunate that he's not on this call because I think that these procedures that are variously known as protein misfolding cyclic amplification, and in his case, the diagnostic assay that he produced, RT-Quick, present major opportunities for future testing. There are two major tests that have been approved right now. One is ELISA, and the other is immunohistochemistry, the latter being regarded as the kind of the gold standard. The ELISA assays are rapid, but we need more sensitive and more rapid diagnostic tests, and this really was a major theme of the working group. And I know that the USDA is actively looking at this, as a diagnostic opportunity, but we're still not there. I think I'm going to be echoing some of the major points that were made in the previous talk, and that is, the opportunity for spillover is a major concern. And in addition to being able to test for prevalence in natural hosts, the notion that we can, conveniently and unequivocally diagnose spillovers, should they happen in other species, including human beings, is a major challenge. Experimental models are getting there, in particular, mouse models that are genetically engineered to express different forms of the prion protein from different species. But these are these have their own challenges, and they're by no means rapid assays. So next slide. So a major issue here in all of this, the umbrella of prion diseases, is the notion of strains. And I've already told you that, like conventional agents, there is strain diversity in prions. So how can you have strain diversity if you don't have an informational nucleic acid? So this is another unusual and unique feature of these diseases. It appears as though mechanistically the strain information is somehow encoded within the conformation of the protein. So this concept it seems to be a good working hypothesis, but it presents major drawbacks to distinguishing strain differences, and the diagnostic tools that we have right now are not good at, if there is strain diversity out there, rapidly and unequivocally determining strain variability. So we know that strains can evolve in wild settings. Whether or not strain evolution is occurring in North America is an open question. And equally concerning is the emergence of new forms of chronic wasting disease in foreign countries, in particular in Northern European countries, Nordic countries more specifically, where the strain properties of the chronic wasting disease agent, we know from experimental studies in various groups, including my own, are completely different to the strain that we know and understand that is causing contagious transmissible disease in North America. So these are some of the challenges that we identified in our working group. The development of new, more tractable, facile, rapid diagnostic assays based on amplification techniques, the ability or lack thereof to discern strain differences, and ultimately the challenges involved in identifying spillover strains should that occur in sympatric animals and also human beings. So I'll stop there, I think.
Okay, next slide. You have this last slide here, Glenn.
I pretty much covered everything.
Okay, good. Okay, we'll move on. Thank you very much then. So, okay, moving on now to the Animal Health Working Group. We have with us Dr. Sabina Gilch from the University of Calgary, where her lab focuses on CWD strains, the factors that influence transmission and diagnosis. This group was co-chaired by Dr. Jason Bartz at Creighton University. So, Sabina, thank you so much for being with us.
Okay, thank you, Mike. So it was really like an honor to be part of this important project. And I'd like to also thank my fellow co-chair, Jason Bartz, and all our working group members. So as you can see on this slide, we also had very broad international representation in our working group members. We have members from academia. We had also members representing the USDA and the CFIA in Canada. So the topic of our group was to discuss challenges around surveillance, laboratory capacity, planning, and response if a spillover event of CWD to non-cervid production animals, such as cattle, sheep, goats, or pigs occurs. And can I have the next slide, please? So that this might happen is a quite valid concern. Because CWD is really a disease at the livestock-wildlife interface, as illustrated in this image. If you drive around, you can frequently see deer and elk using the same pasture, co-grazing with cattle or with sheep. providing exposure to non-survey production animals, either through direct animal-to-animal contact or through CWD prion contaminated environment. Well, I like to point out at this point that naturally occurring transmission of CWD to non-cervids has not been identified to date. CWD has been experimentally transmitted to cattle, sheep, and pigs, and now there is evidence that suggests that CWD can infect wild boar or feral hogs, which might serve as an additional vector for changing CWD strain properties and change the host range of CWD. In general, CWD spillover to non-survey production animals is of particular concern because of potential economic, food safety, human health, and environmental consequences, as we experienced it with the BSE crisis in the 1990s and the early 2000s. However, detecting the natural spillover of CWD would be really challenging. So while there is surveillance in place for BSE and scrapie, both in Canada and in the US, there is no CWD-specific surveillance conducted in non-cervid production animals. And the currently used screening tests that were mentioned in the previous presentation, the ELISA test, only gives you a test that only gives you a result that tells you positive or negative, but it cannot distinguish between CWD, BSE, or scrapie. The confirmatory tests would be more helpful in this regard, but again, we have the challenge that we do not know how CWD would present upon natural transmission in, for example, a cow. So there would be definitely a need for testing additional samples to test distribution and do additional follow-up testing if there was a suspect case in cattle. Next slide, please. So overall, disease investigation would include epidemiological and environmental investigations to determine the potential source of infection, identifying exposure to other animals, and also assess the risk to humans. This is also problematic as production animals, most of you might know, are transported to multiple farms from birth before they finally reach the slaughterhouse and are slaughtered. And so what this means is that the farm where a potential spillover event might have been detected might not be the actual farm where the animal was exposed to CWD and where it got infected with CWD. So investigations would require tracing animal locations and collaborating with producers to sample properties at locations across the country. So multiple agencies then would have authority over the investigations, and there are no plans in place at the moment to outline how different agencies would collaborate, cooperate, and communicate about such potential spillover events. While there are a number of academic centers and laboratories that conduct research on CWD and would be able to offer complementary expertise and models to help the investigation and advance the generation of results, their role in a CWD spillover event is undefined, partly because of the consequences if data are shared that a spillover event might have happened. So the data sharing, I think, is a big challenge here in involving academic laboratories. The next slide, please. So eventually, regulatory authorities would be responsible for interpreting the results of investigations. However, they need to be cautious to avoid any unnecessary repercussions on trade. And we know that there are already impacts of CWD on trade policies. For example, Northern European or Scandinavian countries currently restrict the import of hay and straw from CWD endemic areas. However, there is a need to plan contingencies for the impact of CWD management on trade policies and on the impact of production animals and agricultural industry. It also needs to be considered that, for example, scaling up of testing for CWD in response to a spillover event is different, even though there is precedence that testing can be scaled up for viral infections like COVID-19 the pandemic has shown that capacity. CWD again is different because we have a different requirement for space. We have a lot of unknowns about what test would be best suited and how sensitive and specific those tests would be. So it is again posing different challenges than the viral infections, where upscaling has been done pretty successfully. And with that, I'd like to thank you for your attention and turn it back to Mike.
Thank you, Sabina. Very nice overview. Now we're going to move on to the Disposal and Environment Working Group. We'll hear from Dr. Debbie McKenzie, who recently retired from the University of Alberta, where her research focused on prion diseases and the properties of prion strains. This group is also co-chaired by Dr. Shannon Barteld Hunt from the University of Nebraska at Lincoln. So I'll turn it over to you, Debbie. Thank you.
Hey, thank you. So this working group has had a great mix of academics and people with expertise in various aspects of disposal. So I must say that I learned a lot just listening to all of these people present their views. Next slide, please. So as been alluded to previously, CWD can be secreted into the environment or be present in the environment. We know that infected deer and elk secrete CWD prions in urea, saliva, blood, feces. Also, any carcasses that remain in the environment, on the landscape, are also going to serve as a source of prions in the environment. And we and others have shown that CWD can bind to soil and soil minerals. It can remain there for various lengths of time. We don't have a good handle yet on how long CWD prions can exist in the environment. The binding and persistence is dependent on climate, on soil type, probably on prion strain, although we don't have a lot of evidence for that yet. So this makes it very difficult to know how to deal with this issue. Next slide, please. So our group talked a lot about how we would dispose of carcasses, and sort of carcasses at a couple of different levels. How do we get rid of CWD infected carcasses? As Mike mentioned in our pre-webinar discussion, there's a lot of dead deer on the side of the road. These may potentially be infected animals. What do we do with these? How do we deal with them? As we identify carcasses or animals that are positive after hunting, what happens to the carcasses? What happens to the gut piles? All of these are likely containing infectivity and contaminating the environment. If we have a spillover, now we would have to deal with potentially with carcasses from the spillover, be it cattle, sheep, etc. And we know from experience with the BSE crisis in the UK that dealing with carcasses was a huge, huge issue. In that situation, cattle carcasses were incinerated, and it took forever to process them all. And then if we were to have a spillover to humans, this would probably then add a whole nother dimension to carcass and contaminated item disposal. It's one thing to have a landfill in your community where there's infected deer carcasses, but if there's no risk to humans, people are probably less concerned than if it was to be shown to be zoonotic. So all of that adds to a sort of a critical point in how do we deal with this. We talked about a lot of different disposal methods, landfills being one, incineration being another. You can also dispose of infected carcasses with alkali hydrolysis, but none of these are particularly scalable. So being able to deal with a few animals is possible. Needing to scale up to deal with hundreds or thousands of animals is beyond the current capacity that we have. And since we often have a lag time between when animals are tested and when we know the test result, we then have to begin to assume that all animals at the time of carcass disposal are positive. And that again then adds to the dilemma of how best to get rid of these. Next slide. So we really need to develop a way to assess the effectiveness of the current disposal practices, and this will require a lot of work with the different agencies and groups that currently deal with contaminated carcasses. Landfills are managed by a variety of different people depending on the jurisdiction. So some are privately owned, some are state-owned, and so working through all of the ramifications of this is complex. And as I've already mentioned, any spillover would then change the entire disposal process, potentially generating a lot more waste that we have to manage. And again, if there's zoonotic potential, changing the entire perspective of community members as well. And with that, I will throw it back to Mike.
Thank you very much, Debbie. Very, very thoughtful overview. Next, we're going to move to the Human Health Working Group. And the two co-chairs were Dr. Brian Appleby and Dr. Lawrence Schoenberger. Dr. Appleby, who is currently the National Prion Disease Pathology at the Center and Case Western Reserve University, and a foremost authority on human prion-related diseases, will be presenting the Human Health Working Group summary. Brian?
Thank you, Mike, and thank you to you and your team at CIDRAP for putting together this much-needed report. I want to thank my co-chair, Dr. Larry Schomburger at the CDC, as well as everyone in our working group. As you can imagine, it required a lot of people that were clinicians, state public health officials, CDC, and people that unfortunately had also worked on prior zoonotic transmissions of prion disease in the form of bovine spongiform encephalopathy to humans. Next slide, please. In humans, CJD is what we call naturally occurring prion disease. It can occur sporadically through a genetic mutation or can be acquired either through transmission between individuals or zoonotically with bovine spongiform encephalopathy. So we do have precedents for this. There is a concern that chronic wasting disease could spill over into humans. We have precedents for this with bovine spongiform encephalopathy. And before, we did not know what that would look like as well. Luckily, with bovine spongiform encephalopathy in humans, which we call variant CJD, these were unique individuals that came down with prion disease. They were younger, the neuropathology looked different, and even their clinical diagnostics were slightly different, which allowed us to detect an unusual new form of prion disease that we could then tie back to BSE. However, we don't know what would predict a current or future spillover risk. We know that there are some animal prion diseases that have not spilled over to humans and some that have not. So we don't know the exact risk, but we know that it's not zero. There are other things that also will affect our determination of what that risk is. As you heard earlier, there's different strains, perhaps within the US, but definitely across the world. Those strains might have different abilities to transmit to humans or other production animals that they then may be more likely to transmit to humans. And unfortunately, in prion disease, as many of you are aware, we have a very long incubation period. So even the human transmission incubation period is measured in years. The only zoonotic illness that we know with variant CJD, the mean incubation period was about 10 years. So it could be that chronic wasting disease is transmitting to humans already. We just don't know it because the incubation period is so long. And it's also very difficult to apply experimental results to real world data. In the lab, these are very controlled environments that we're able to do genetic manipulations and exposures in very specific ways. However, that does not happen in the real world. Things like dose, exposure, it is very variable, and we have no way to know that and put an experimental limitation on that. Next slide, please. Doctors are getting better at recognizing human cases of prion disease. As you heard earlier from Glenn, there's a new test called real-time quaking-induced conversion, which allows us to amplify minute amounts of the prion protein in order to protect it. However, one important thing with that test, although it's completely revolutionized the human diagnosis of prion disease, is we know that it can be variable, that there are certain strains that may actually not be detected in this manner. And the concern is that perhaps if chronic wasting disease were to spill over to humans, that it may not be detectable in that manner. We also saw this with variant CJD in humans, and that even brain MRI profiles were different in cases of variant CJD compared to classical sporadic forms of the illness. The way that we address this in the US and in other countries that have surveillance systems is we conduct autopsies of cases suspected of having prion disease, because the pathology does look different dependent on the type of human prion disease, but also if it is a zoonotic spillover for variant CJD. The assumption is that because this is a novel prion disease, we would have unique pathology on a human that was transmitted with chronic wasting disease, but that's not guaranteed. So if it were to look like a case of sporadic CJD that we already know and are familiar with, that would be extremely difficult to determine and would be very difficult to determine one case. We would have to require on epidemiologic evidence in order to detect the likelihood of prion disease. Unfortunately, as you've heard from pretty much all the other speakers on this call, we haven't really defined what the evidence is that would identify a chronic wasting disease spillover incident. And that's because we don't know what it would look like. Chronic wasting disease prion looks very specific in a cervid, but because it gets transmitted to a human and the human prion protein is the substrate for which it amplifies, we don't know what that would look like from a prion standpoint, neuropathologic standpoint, or clinical standpoint. So we're highly reliant on things like novel neuropathology as well as epidemiologic evidence, such as exposure or differences in demographics of people that are developing human forms of the disease. Next slide, please. So in summary, one of the things that we would really like to be able to do is get as much exposure data as possible. However, this is not always possible. We do collect information from families who are kind enough to donate their loved ones' brains to the Prion Center regarding things like venison consumption as well as hunting history, but that may not always be accurate. We also don't know many times whether or not venison consumers or hunters have actually been exposed to chronic wasting disease. So CDC and state public health departments do have certain projects that focus specifically on some of these risk factors, but largely it's unknown. And of course, the spillover collaboration is somewhat limited by things like buy-in, data privacy, funding, and spillover unknowns. There's the CDC as well as state public health departments. But the way our healthcare system is run, we don't usually have easy access to healthcare data to be able to do good epidemiologic research on clinical data, as well as declining autopsies across not just the US, but across the world, also threatens our ability to detect novel forms of prion disease. So with that, it's very important that we continue to be vigilant about human prion disease in our country to make sure that we don't have novel forms of the disease, especially potentially chronic wasting disease in humans. But as you've heard, there are several limitations to that. And with that, I'll turn it over to Mike. Thank you.
Thank you, Brian, for a great overview, and to you and Larry both for co-chairing this. Let me now just summarize a bit for you with the report conclusions and the recommendations. We took the key findings from each of the working groups and came up with 9 recommendations for how to address gaps and the needs of the field. Next slide, please. The first one is to advocate for CWD spillover awareness and preparedness among medical providers, research institutions, and wildlife, agricultural, and public health agencies. Number 2, to create sustained, dedicated, multi-year funding sources to support the needed efforts. The third one, expand and standardize CWD surveillance in wild cervids, other wildlife, and non-cervid production animals to facilitate data summarization and interjurisdictional comparisons. And number 4, promote human prion disease surveillance among human healthcare providers to enhance the capacity to detect CWD spillovers in humans. Next slide. No, also, number 5, to expand CWD research and development efforts. You heard that loud and clear, I think, through every one of the presentations today. Number 6, encourage and support formalized interagency and cross-sector collaboration, something that is going to be critical at a time if there ever should be a potential for a spillover event. 7th, develop and test proactive CWD spillover communications, messaging, and education well in advance of any possible future challenge. Number 8, draft regulatory and governmental agency policies in advance. And finally, plan for environmental concerns and estimate disposal capacity needs. These 9 conclusions and recommendations really summarize what was many, many, many hours of work and dedication to this. And we believe these all still are very, very important even today. Next slide, please. Before I go on, I just want to note the incredible team at CIDRAP who've been working on the CWD effort for several years now. Co-directors Jamie Umber and Corey Anderson, and program managers and researchers Leah Moat and Claire Stoddard, and our news editor-writer who has been really keeping the world updated on CWD activities for our website, Mary Van Busikum. I want to thank them. This could not have been done without this group. You were outstanding. So let me now move to the next slide, in which we get to the interesting part, you might say, of the give and take here. Now we're going to open up to questions and answers, and Corey Anderson is going to moderate this. He's been following the chat. I see we have a number of questions or comments in the chat already. We'll bring all the speakers back up again on the screen here so you can see them. And at this point, we'll go ahead and turn it back to you, Corey.
Yeah, well, thank you. And obviously, thank you to all of our speakers. I think that was a great overview. And of course, if people are interested in learning or reading more, we have the report on the CIDRAP site, so we'll make that available. As Mike said, too, a lot of really great questions. I've been going through all of them, and fortunately and unfortunately, we've had some related to the same topic, just sort of different takes. So I'll do my best to get to them and maybe have to combine some questions. And so I guess I'll start out. Glenn, this might be up your alley here, although I know we have a couple of prion biologists on this call, so feel free to chime in as well. But we did have several questions related to the whole issue of CWD strains or prion strains in general. A few are a little more technical, so I guess I'll just pose to you, Glenn, first. I mean, if you had 1 to 2, takeaways that you'd like attendees to sort of leave this webinar understanding related to the issue of CWD strains, what would those takeaways be if you had to really boil it down when it comes to CWD strains? Maybe what's not necessarily understood that you'd like to sort of put out there.
I'd re-emphasize the unusual biological nature of the actual infectious agent. We've all lived through COVID recently, and we all became more aware of the emergence and transmission of variants of the COVID-19 virus. And we all, at some level, understood that this was due to the fact that the mutation, the nucleic acid mutation rate in these viral populations was high and error prone, and this led to natural evolutionary tendencies and by natural selection the emergence of increasingly fitter variants. The fact that this doesn't happen, it is a fact, with prion diseases is a revolutionary idea. However, we know that strains do exist. There's a rich body of evidence, experimental and natural settings that underscore this concept. So the $64,000 question is, how does strain variation occur? And so this is an active area of research in the field, and only recently has it become possible, due to the work from Byron Coey's lab and also John Collinger's lab at the MRC unit in London, to gain some high resolution insights into the molecular basis of this. And it's clear now that different strains have different conformational shapes that somehow encode or encipher this strain information with a high degree of fidelity from one generation to the next. Not only that, but we know that strains evolve in the same way that viral strains evolve because of natural selection. Uh, but how does this also occur in the absence of a nucleic acid? And there's a wide body of evidence showing that strains can and do evolve both under experimental and natural settings. Strains conventionally have been thought to evolve most readily by transmission across a species barrier. So these two issues are really intimately related. The variables controlling the extent of a species barrier, in other words, the ease or difficulty with which prions from one species can cause a disease in another one, and how strains impinge upon that process. I think there's an emerging picture, and to some extent this hasn't been published, but that the strain profile of CWD in North America is, for all intents and purposes, with some few exceptions of variation, pretty stable. This mirrors our experience with BSE. There was one dominant strain that was causing globally bovine spongiform encephalopathy in cattle. And there became clear that there were rarer variants that were also emerging as surveillance was occurring. I think, from my perspective, and this is, as I said, this is more or less unpublished work, but it's becoming clear to me as an expert in the field that there is a major single dominant strain that's responsible for contagious transmission in North America. But the issue is that we don't know the evolutionary properties of that strain, and these evolutionary events can and do occur over very long time frames, and this raises the ante then for the potential for novel strains to cause disease and have expanded zoonotic potential. So I'll stop there.
Thank you. I appreciate that. And like I said, other panelists, I know we sort of have some expertise across the board here, so always feel free to chime in as well. I guess next question. This was a good one, and it's aimed at Brian. But an attendee had a question that said, given the risk of CWD spillover to humans, or given that the risk of CWD spillover to humans is hypothetical, how would you encourage and engage departments of health to participate in interagency CWD response? And I guess if you want to even take that a step further and just speak to, your perspective when it comes to, cross-disciplinary response and that status and where we stand in the prion disease world at this point.
Yeah, I think state public health departments are extremely useful, especially for use case scenarios like this. The way it stands now is CDC does national surveillance, and then states, or some of them are funded by CDC to do extra surveillance for prion disease. However, prion disease is not a mandatory reported illness in all states. So that can be somewhat of a difficulty and also somewhat inhibits our ability to get the data that we need. States can be very helpful. We do share all our data with state public health departments so they know of suspected cases, and hopefully they reach out to patients' families and can investigate whether or not it's a true case or a false case, as well as encourage the family to pursue or at least consider postmortem for brain analysis. And they can also be exceptionally helpful for getting the information that we need about possible exposure risks. With that, I think it would be very important for them to be very close to agriculture and wildlife within their states as well, to know what the status of chronic wasting disease is in their state, what resources are available for testing, the likelihood that individuals that they're investigating could have been exposed.
Perfect. Thank you. This one I'll just pose to the group as a whole, so feel free to chime in with any thoughts. But we did have a couple of questions related to just, where do we stand as far as understanding, the risk of potential spillover, whether that's to humans or production animals. And so I guess I'll broaden that to, um, what's everyone's overall take, or if you have a strong opinion, what's your take on just the current landscape of research that has assessed the risk of interspecies transmission? How confident can we be based on the work that's been done to date? And I guess if you want to build off of that, what directions do you think, in your opinion, we need to go, or what are future needs in that area?
So maybe I'll just begin by saying, to me, the discovery that chronic wasting disease has transmitted seemingly very efficiently to feral hogs in areas of Texas where rates of chronic disease, it's an epidemiologic study largely, but where chronic wasting disease occurs and animals coexist, there's evidence that they can develop signs of disease is extremely concerning, because this would suggest that this is a relatively facile process. I don't know if there are other comments from the group about this or other issues relating to interspecies transmission, but I find that quite remarkable and concerning.
Thanks, Glenn. Any other thoughts from the group?
I think that some of the work out of Justin Greenlee's lab, USDA and Ames, has also suggested that CWD can move in to other species, such as raccoons, and again, potentially changing the host range. Um, a little bit more distant from that would be studies using transgenic animals. We've done some work with transgenic mice expressing beaver PRP, and we can, in the lab, infect those beaver mice with CWD and with relatively short time frames. Transgenic mice are a long step away from a natural infection of beavers. But again, these suggest that we need to be broadening our surveillance as we move forward.
Thanks, Debbie.
Yeah. Maybe I can add on like studies that were done to investigate the potential transmission of CWD to livestock. So a lot of this work, as Debbie just mentioned, was done at the USDA by Justin Greenlee to look at transmission to cattle, sheep, and pigs. And while, for example, in cattle, there was indication that transmission might be possible. Even like the models and the transmission modes have some limitations. So they were mostly susceptible if CWD prions were injected into the brain, which is not a very natural route of exposure. And I think there is still like a lack of understanding at what needs to happen to get CWD infection under natural settings. If those animals do not develop clinical disease, would we need to be worried about subclinical disease, which might have happened in those feral hogs? So they have signs of infection, but they are not actually clinically sick. And I think these such animals in a subclinical stage, like being infected, propagating prions, but not developing clinical disease, are a huge concern as well, because they might potentially disseminate and transmit the disease. So we're looking at different pictures. At the end of the day, clinical animal that dies of prion disease is easier to handle because we can recognize it, why we might have animals that don't get clinical sick, but contribute to evolution of strains and transmission of the disease.
Thank you all. That's great. Let's see, another couple of questions that are related. And Brian, this is again probably more aimed at you. But just getting into this idea of potential spillover to humans specifically. One person asked just how likely it could be that human cases of CWD may end up being confused with dementia or other neurological diseases. I guess another person, somewhat related, talked about this idea of, is there value in looking at organs other than the brain in suspected or potential cases to evaluate spillover risk? So if you could speak to those, that would be perfect.
Yeah, and just to follow up off your last question, I think we all can appreciate that if chronic wasting disease does transmit to other animals, and then we find chronic wasting disease transmits to humans, that's going to pose another challenge as far as what the vector is, where is it coming from? And I hope we don't become in that scenario. I think it is helpful to look at other organs, and in fact, we are doing that at the Prion Center. We know that in chronic wasting disease, it does seem to be somewhat lymphotrophic. And we also know that if humans were to develop chronic wasting disease, it would most likely be from the oral route. And like variant CJD prions did reside pretty heavily in lymphoreticulaire tissues. And of course, that became a concern for our blood supply. So we are also collecting lymphoreticulaire tissues in people that are highly suspected of having human prion disease and at least have some possible exposure, whether it be venison consumption or hunting in a CWD endemic area. Again, we don't know what chronic wasting disease in a human would look like. We do know that for the most part, all human prion diseases are, well, they all are fatal, but for the most part, they are also rapidly progressive. So it would be our hope that anyone who has an atypical, rapidly progressive dementia or degenerative disease would come to autopsy. And we have, tried to reach out. In fact, we were at the American Academy of Neurology this week to encourage physicians to alert families about our autopsy program, even if it doesn't exactly look like prion disease, but is a unknown rapidly neurodegenerative illness. Good question.
Thanks, Brian. I guess we'll turn it to Russ and get back to some more wildlife specific issues with this topic. But I think, again, a number of questions related to this idea of, the risk of potential spillover to potentially, scavengers. Someone mentioned, specifically, are we testing feral swine for prion disease? I guess, Russ, I'll turn it over to you when it comes to, CWD surveillance that wildlife agencies are doing. I know we've touched on it a bit throughout this webinar, but is there anything you'd like to add just that might give some perspective to folks who are curious, what's the current status of surveillance in cervids, maybe potential, limitations or gaps there? And then can you just speak to, prion surveillance in non-cervid species and where we stand with that?
Sure, Corey. Well, let's start with prion surveillance in cervids. And as I indicated, it's somewhere from none to tens of thousands, probably depending on resources or people running programs. But again, there's no standardization. I can think of one instance between two states where political machine in one state was criticizing the agency because the state next door had no CWD. In that case, it was because the state next door wasn't doing any surveillance. And of course, it's hard to find things if you don't look for them, although occasionally I find that to be untrue. When you talk about scavengers, it's probably a mixed bag. There are some who believe that scavenge will clean up a CWD infected carcass, and that's a good thing. Probably is, might be. We don't know how many of, whether that site is actually clean. But then there's another piece that's important to recognize, which is to say, what goes in comes out. And as a consequence, those predators or scavengers could be moving CWD around the landscape. And that becomes important because most herbivores are coprophagic, which is to say they go around and eat, they're going around eating waste from other animals. Rabbits do it, deer do it, lots of herbivores do it. And so the potential exists for predators to actually move CWD onto landscapes where it isn't previously been detected or was previously non-existent. The data aren't there. When it comes to looking at agencies' surveillance of species other than cervids, it's non-existent, really. What Glenn was talking about, what I mentioned, was a study that was conducted by USDA Wildlife Services in association with folks in Arkansas and Texas. But it's not part of anybody's routine. And that's potentially important. In Texas, for example, one way of dealing with feral hogs is to corral them, essentially purge them by giving them clean feed and then selling them into the human food chain. Likewise, behind high fences, you've got Eurasian boar being a mainstay of the captive cervid industry. They sell deer, but they make their livings on hogs. Nobody's paying attention. Is that potentially a source of transmission between high fence operations? There's a lot of work to be done there. And frankly, at least from my perspective, I don't think it would be an overstatement to say that nobody's looking.
Thank you, Russ. Appreciate it. And I might actually keep you in the hot seat while I have you here. Another question that I think is a good one. They raised the point, it's sort of in their question, but I'll just maybe answer it for them in the first part and say that obviously, prevention of the spread of CWD is important. Management of CWD and wild and captive cervids is obviously important as well. I guess in terms of, spillover, based on the discussions that the wildlife group had, I guess could you just briefly go over, what might be the consequence or implications of confirmed spillover of CWD and what impacts that might have on wildlife management as we know it now? Like how could that change, or what should we be thinking about in terms of that?
Well, Mike called me an expert, so I guess I can put on my expert hat and pontificate my perspective. My perspective on this issue is that it would be catastrophic to agencies for several reasons. First off, somewhere between 60% and 70% of all license revenues going to state fish and wildlife agencies comes from the sale of deer hunting licenses. So it's the mainstay. And it's important to recognize that 80% of all agency funding in the United States at the present time comes from the sale of hunting, fishing, trapping licenses, and associated excise taxes that are provided by the federal government. So the loss of that revenue would be absolutely catastrophic to conservation of all kinds for game species, game species, imperiled species, and so forth. But there's another piece as well, which is to say, if it were to spill over, other agencies will take priority, I guess is another way of putting it. If it's harming the human population, suddenly human health agencies are driving the show when it comes to deer management. If it spills into agriculture, agriculture becomes the one that's driving policy. And so, as Mike mentioned, it becomes very important to think about those relationships among agencies right now, so that if it ever does occur, there's some coordination, because there will be fistfights between the jurisdictions, because that's what agencies do, and it's probably best to have those disagreements before the problem actually occurs.
Thanks, Russ. Appreciate that. Another one that might be more relevant to the prion biologists among us here. Again, going back to this issue of strains, I think I just want to combine these questions and just ask for maybe summaries from you if you have strong opinions of just explaining, how easy or difficult is it to differentiate between strains? What do we sort of need to improve our understanding of strains? Just I'll leave it at that for now. Basically, I just want to get into this area of, is strain typing of prions, where do we stand when it comes to that?
I'll jump in on this one first. Sorry, Glenn. I think that we are still actually at one level struggling to define strains. There's very classical definitions of what a strain is, based back on strains of sheep scrapie, where transmission properties, incubation periods, neuropathology defined a particular strain. But as we start looking at them with more biochemical and molecular tools, um, depending on what tool you use, you can see different things. So we have strains in the lab where if we look at them with one particular set of tools, we can differentiate them into two different groups. But then when you look at them in terms of transmission characteristics, they're exactly the same. So I think that a lot of the definition of strains is going to get reworked. There's clearly differences in their biochemistry. How important is that to the biology? I don't know if we've got that delineated. Glenn, Sabina?
Sabina, you want to?
Oh, you go first. You have your map.
Yeah, I mean, strains are pretty much operationally defined, right? And the other point about this that Debbie alludes to is that their properties change based on how you analyze them. It's a very challenging circular issue. So, for example, if strains are classically defined by their transmission properties into susceptible hosts, that's how we define strains, and we have classically over the years. But it's no secret that depending on how you transmit the infectious agent to the actual host, in other words, whether it's directly into the brain or via the periphery, you can get different outcomes. So what that tells us is that the properties of strains are malleable. It relates back to this issue of evolution. From a molecular perspective, it's even more complicated because it appears as though there are a strain is it's kind of like whack-a-mole, right? It's not one stable collection of infectious agent, but rather an amalgamation of different possible states. And there's a dominant conformation, if you like, that can exist under certain circumstances. But when you change those circumstances, those properties can also change. So it's a very, very complicated biological problem. It's not that this concept isn't unique because it also applies to viral populations. We're talking about what's called biologically quasi-species. It's not unique to the prion world, and in fact, it's a concept that's been borrowed from viruses. But it makes it very difficult to define strains and categorize them. And yeah, I'll leave it at that.
Yeah, I mean, I agree with what has been said, that it's very difficult, or basically at the moment we don't have defined criteria for strains. But with regards to CWD, I think at the end of the day, what is relevant is strains that can cross species barriers, that can infect species or insert genotypes that are resistant or less susceptible to what Glenn alluded to, the probably major strains circulating in North America, because animals adapt to CWD infection. We see an increase in those more resistant genotypes, for example, in Wisconsin and deer. But then the agent would adapt to that as well. So we have evolution of strains. But I think at the end of the day, the focus, if we identify new strains, should really be looking at, is this strain possible, able to cross species barrier, to infect humans, to infect non-cervid species?
Excellent. Thank you all. And I know we're getting fairly short on time here, so I'll pose one more question and pose it to the group as a whole, so feel free to chime in. But I guess thinking about CWD as, an emerging disease, with obviously a lot of unknowns, I guess for the group, what lessons do you think we can learn from other prion diseases that we are aware of and familiar with, that can maybe be applied to better prepare ourselves for a potential spillover scenario with CWD? Are there reassurances that we can take based on other prion diseases? Are there things that, we should be concerned with specific to CWD, maybe differences? Any thoughts would be perfect.
So maybe I'll take a stab at that. I mean, I think your use of the term emergent is really important. This is an emergent disease, and the latency period for infection is extremely long amongst all of these prion diseases or transmissible spongiform encephalopathies. So we're talking about time scales that of many decades in order to really understand the potential for emergence of new strains. I would also point out that in recent decades we've lived through the BSE crisis. We got pretty lucky with BSE. This was a single unique strain that was transmitted in a foodborne way and was not contagious from animal to animal. I'm dramatically oversimplifying things, but basically, if an animal got infected as a result of feeding, other bovids in herds were not highly at risk for contagious transmission within those herds. This is not the case for CWD. This is a highly contagious, transmissible disease, and it's the only disease that we know of that affects not only captive livestock animals, but also wild animals. So the challenges surrounding control of this highly contagious disease of wild animals are enormous. And superimposed on top of that, the unknowns of evolution, and the fact that this is almost uniquely an inevitably fatal disease. You don't get well if you're infected with prions. So the stakes are pretty high here, and the uncertainties are not minor.
I would just add on to what Glenn had all of my points pretty much, but the fact also that CWD is present in the periphery of deer, so it's being shed into the environment. We don't understand the role that the environment plays. In the BSE crisis, the BSE prions were pretty much localized to the central nervous system. So as Glenn said, no animal-to-animal transmission, no transmission into the beef farms or dairy farms. So it kind of minimized the exposure. We just don't have enough information about where CWD is to understand what the ramifications are. And I think the other key point is that the range of CWD in deer continues to expand. I think we're up to 36 US states and 4 provinces in Canada. And the prevalence within a given herd is increasing, so that for example, an area of Saskatchewan, 85% of the mule deer bucks are positive. So this is just a huge exposure risk.
Also, it's worth pointing out that the host range, not only the geographic range of CWD is expanding, but the host range, in other words, the number of different species that are infectable by CWD, both in North America and in Northern Europe, is not static. It's increasing. And so I think that gives extra pause for concern.
Thank you both.
I mean,
Well, I think with that, yeah, I'll pass it over to you, Mike. Thank you all again.
yeah. Well, first of all, I want to thank our speakers today and for your work as co-chairs, which was absolutely essential. And I also want to thank the other co-chairs who are not with us today, all the members of the 5 working groups. You can imagine, if you are not a member of the working group, but listening to this conversation today, the intense nature of learning that occurred throughout this process. And in many instances, understanding what we didn't know and how important it was to learn what our future might look like. So I just want to thank you all very much. I want to thank all the participants today for your questions. Thank you. There were over almost 50 questions that came in. We could have gone on for quite a bit longer. This webinar has been recorded. It will be available on our CIDRAP site and a YouTube channel by the end of the day today. Also, you can see the QR code there to subscribe to the CWD newsletter from CIDRAP, free of charge. We're not selling anything. Hopefully you find it helpful and useful. And again, thank you so much for this participation. We will continue to pursue our work here with CWD the best we can with furthering your work. I think one of the things we see ourselves for now is an advocate for many of the people that are part of these work groups, and clearly our speakers today, advocating for them so that they have the resources they need to deal with this. So again, thank you. I also want to give a specific shout out to Russ Mason, who not only is he serving as the expert wildlife person, but he has been a consultant to us as CIDRAP on this project. And his input has simply been invaluable. Thank you, Russ. So with that, we'll close, and thank you again very, very much for attending.